This review is for the Biology Keystone exam. We will be covering questions 21 through 24 in our Biology Keystone review packet. To start off with, let's talk about why do cells divide in the first place? Why go from one cell to two cell? There are really three main reasons that we can talk about. Cells need to simply get bigger, and organisms need to get bigger. You have millions and millions of cells in your body, but you started off as one teeny tiny cell. So by simply dividing, you have helped you get bigger. We also have the idea of repair. If you bump your arm or scrape your leg or something like that, you need to replace the cells that were damaged. So simply dividing helps replace them. And then we have reproduction. Asexual reproduction, very easy. I have one cell, splits into two. Done. For organisms that reproduce sexually, it's a little different, and we will cover that division process as well. There are a kind of set pattern or set number of steps that we go through in our eukaryotic cell division cycle. Remember, as a eukaryote, we are an organism that has a nucleus. So we, trees, those kind of things. Prokaryotes like bacteria, they don't go through this cycle. So you can see there's just kind of this general cycle, and then there's this little piece here. There are certain times or certain types of cells that will go into maybe this little extra piece called G0. And G0 would be a time of cell arrest or no more growing. And you can see here, really good examples of this would be brain cells. So glial cells and neurons are both types of cells in your brain. They simply don't go through this cycle. However, if we're looking at the normal cycle, we're looking at something like this. And you see the intergrade cycle, the biggest chunk here, is a part called interphase. Interphase is really all about preparation, while this little teeny tiny sliver, that's mitosis. That's where the actual dividing happens. We take interphase and we break it into three parts. We have a G1 piece, an S piece, and a G2 piece. And then our mitosis piece, or this M phase, we split into four parts. We have a prophase, a metaphase, an anaphase, and a telophase. And we're going to talk about all these in a little more detail. So the first part of interphase is called G1. And I remember this as G means growth, you're just growing. That's it. When the cell divides, you can see it goes from this big to a much smaller size. So to prepare to divide again, it simply needs to start growing. After our growth phase, we have our S phase, or synthesis. We're synthesizing new DNA. We're duplicating our genetic material so that when we are finished this phase, we will have two pieces of genetic material, one for each of the new cells. Basically, the idea is I take my original strand or piece, I split it in half, I add in some new nucleotides, remember those are the monomers of DNA, and then I make a second piece. We're going to go into this process in much greater detail in a later podcast, but right now I just want you to kind of look at one thing. You can see we have those four different nucleotides. We have guanine, adenine, cytosine, and thymine. And the idea is that they match up with certain ones. So G always goes with C, A always goes with T. So if I were to split a strand in half, I can recreate the other half simply by looking at the template. So if I see a G, then I know that I'm going to have to put a C in its place. If I see an A, I know I'm going to have to put a T in its place. And that's how we can conserve the genetic code through cellular division. After our S phase, we have G2, which I think is growth times 2. It's the second time the cell is trying to grow. It's also like a little checklist. Do I have enough mitochondria? Do I have enough cytoplasm? Do I have everything I'm going to need in order to go ahead and divide? Once that checkpoint is kind of established or met, then we flip into mitosis. And again, mitosis is all about dividing the contents of the nucleus, and we have four stages. We have prophase, metaphase, anaphase, and telophase. Telophase also has this other stage that happens at the same time, cytokinesis, and we'll mention that when we get to telophase. But right now, I just think PMAT is how I remember it. Prophase, metaphase, anaphase, and telophase. So a quick overview looks something like that. There's a lot going on there, so let's break it down a little bit. The idea is when we go through this whole funky process, we want two cells that are exactly genetically the same. I have 2N to 2N here, and that N just means a set of chromosomes. So in your body, Your magic number is 46. You have 46 chromosomes in every single one of your cells. You have 2 sets of 43 chromosomes in every single one of your body cells. So what happens is if I hurt myself on the tip of my pinky, I'm going to go through mitosis, so I have identical pinky cells, identical skin cells, 2 to 2. In addition to this 2N word, it's sometimes referred to as diploid, and it's just, again, referring to 2 sets. So I think of head, shoulders, knees, and toes, that song, those are where your diploid cells are. They are your somatic cells or your body cells. So just a little idea here, leading into interphase, that last little bit of it. Prophase looks something like this. We have metaphase down here looking something like this. Anaphase looking like this. But what you can see is they have genetic material that's doubled, and the idea is kind of to try to split that genetic material evenly between 2 cells. So our first stage is prophase, and I think about prophase as the preparation phase. We have a nuclear envelope that surrounds a nucleus. During prophase, that starts to disappear because you want to get to this stuff in the nucleus. You also have your centrosomes, and your centrioles were what these were kind of consisted of, and then all these little fibers. I know they look like spiders. Those fibers are starting to attach because they're going to help pull away the chromosome. Our DNA gets all nice and super condensed into these chromosome structures so that it's very easy to latch onto one and pull it apart. After prophase, or our prep phase, we have metaphase. And the way I think about metaphase is in the middle. Your chromosomes are lining up along a metaphase plate, or a plate in the middle of the cell. So you can see they're trying to line up in the middle. You'll also notice that these centrosomes, or these spindle fibers, have moved all the way to 2 opposing poles, or 2 opposing ends of a cell. And the idea is that they're going to latch on to these little circles, and then they're going to use them to kind of ratchet and move away. So you can see that better in our picture here. And this phase is called anaphase. During anaphase, what happens is these little sister chromatids, these chromosomes, have separated. You can see an increasing separation, and the cell is getting longer, and we're moving stuff to 2 separate ends of a cell. So anaphase, moving away from each other. And then we have our telophase. At this point, you're pretty much completely separated, and telophase is kind of like the end. It's almost the opposite of prophase. So those spindle fibers that got super long in prophase, they start to break down and get smaller. My chromosomes that got all nice and condensed and strong, stark X's, they're starting to kind of turn into mush again. And then also, at this point, you can see that my nuclear envelope, or my thing that goes around the nucleus, is starting to reform. Because again, unless I'm directly dealing with those chromosomes, I want to try to protect them as much as possible. And to take it a little further, there's also cytokinesis, which looks something like this. And in cytokinesis, what we're looking at is kind of separating the actual cells. But you can see, at this point, they look like 2 distinct cells. Plants and animals have a teeny tiny bit difference during telophase and cytokinesis. Mainly because plant cells have a cell wall and animal cells don't have a cell wall. So in plant cells, what we can see here is during the anaphase, we're all moved away during telophase, what happens is it brings in this thing called a cell plate. And basically what you're doing is you're making a new cell wall to join and to separate those cells, so now they're distinct cells. In anaphase, in animal cells, what happens is at the end of anaphase, we get this little ring of filaments and tubules, and what it does is it kind of wraps around the cell and pulls it in together. It is called a cleavage furrow, and it pulls it on in, and basically pops it into 2 separate cells. Just a little bit of a difference there. McGraw-Hill has a really nice video on mitosis, so we're going to look at that to try to pull everything together.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between 2 daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Okay, so moving on from mitosis to meiosis. Certain times we're going to want to cut our chromosome number in half. Like I said, as human beings, our magic number is 46. So if I have 46 chromosomes and my mate has 46 chromosomes, our child would have 92. 92 is not the correct number for a human being. And then if this person were to grow up and he were to mate, his child would have 184. We do not want this scenario to happen. We want to keep the correct number 46. So what we do is we take this chromosome number and we simply cut it in half. And we do that through a process called meiosis. And for that reason, sometimes meiosis is called reduction division. I want to reduce the number of chromosomes in each cell. So I start off with 2n, or my 46 number, and I cut it down to 1n. I cut that number in half to 23 chromosomes. Again, diploid is referring to 2n. Sometimes it'll be diploid to haploid. So we're going from now half of what we had before. This only happens in your gametes or your sex cells. So this is not head, shoulders, knees, and toes. This is sperm and egg. To have this happen, we actually go through two different sets of division. So we have our interphase, we have our prep. We're going to have a meiosis 1 that's going to result in 2 cells. And then we're going to have a meiosis 2 that's going to result in 4 cells. So at the end, we get 4 haploid cells, and they are not genetically equivalent. In mitosis, they're all identical, they're the same. You can see here half the number and they look different. There's kind of a mishmash of coloring. So meiosis 1, you can see here interphase, not condensed DNA, it's where it's spending out its time. Prophase, prep. Metaphase, I'm still lining up in the middle, but now I have these homologous pairs of chromosomes. So homologous just means matching pair. So since I have double, I have my matching pairs here. They separate and I get to telophase 1. So I have my interphase, I have my prophase 1, I have my metaphase 1, I have my anaphase 1, I have my telophase 1. Really the same idea. I still have my prep, I still have my middle, I still have my away, and I still have my far, because telo means far, or the end, but I'm looking at different amounts of chromosomes. I have big paired homologous chromosomes that are doubled up. So at the end of this, I'm going to get 2 cells, and each is going to be 1n, or haploid, because it's going to have half of the number that it used to have, and it's got 1 set of chromosomes. For meiosis 2, what we're going to have is a prophase 2, a metaphase 2, an anaphase 2, and a telophase 2. You'll notice there is no interphase 2. And that's really important to think about, because in interphase we have that S phase, and S stands for synthesis. We don't want to redouble our genetic information. We want to cut it in half. So in our prophase, what's happening is those chromosomes are going to make sure they're quick little prep, make sure everything's correct. Metaphase, they're going to line up right along that metaphase plate in the middle. Anaphase, what happens is those sister chromatids, the half of the X splits apart from each other, and then telophase, the end, they're moved far apart. This one is almost identical to mitosis. Meiosis 1 is more different. At the end of this, you get 4 cells, 1, 2, 3, 4, and each one is 1n, and it has 1 set of 23. So if this were a sperm and this were an egg, 23 from sperm plus 23 from egg equals the 46 of a normal adult. McGraw-Hill also has a very nice animation of meiosis, so to sum everything up, we're going to watch that one.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only 1 member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only 1 of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
So the McGraw-Hill one just reiterated this, but there's sources of genetic variation in the result of meiosis. You don't look just like your brothers or your sisters, and you don't look just like your parents. And there are 2 main things that happen in meiosis that illustrate this. So one that was mentioned is prophase I. There's this idea of crossing over. And again, what happens is you're matching your homologous chromosomes, and during prophase I, they actually can cross over another and swap little pieces. So what you can see here is this is an AB, a, little b, big B, little b. They're just different combinations, and that allows us to be varied or different. We also have independent assortment, which happens in anaphase 1 and anaphase 2. And that's the idea. During anaphase and during metaphase, you don't know how they're going to line up. You don't know how they're going to separate. So they're showing you that all the blues could be on the left and all the reds could be on the right, or blue, blue, red, red. It's completely equally probability. It's just luck. So that those cells could look like this, and those cells could look like this, and those cells could look like that. So it's really the lining up in metaphase and the moving apart in anaphase. So just to sum up meiosis, we have one cell here going through interphase. We go through prophase 1. Nuclear envelope breaks down. My homologous chromosomes start to pair up. That's where crossing over would happen in prophase 1. Metaphase 1, they line up in the middle. The homologous chromosomes separate, go away from each other in anaphase 1. And then I have telophase 1, where everything's kind of reforming. There is no interphase 2, so we jump right from telophase 1 to prophase 2. Again, prepping, making sure everything's set, get those spindle fibers moving. Metaphase 2, lining up in the middle. Anaphase 2, moving away from each other. Telophase 2, the end. And then when it's all said and done, you get these cells. So this one would have twice the amount of genetic material as these. However, even though this process seems pretty awesome, and it is, it can go wrong. And the biggest way it can go wrong is in non-disjunction. So what happens is, again, you're supposed to line up in the middle of the cell in a metaphase, but what can happen is instead of pulling those two up and those two down, sometimes these homologous chromosomes don't separate. If this were to happen meiosis 1, and what happens is instead of getting these even numbers, you get something like n+1 or n-1. This can lead to genetic diseases like Down syndrome, where you have 3 copies of chromosome number 21 instead of the 2 you're supposed to have. There's also Patau syndrome, which is trisomy 13, so 3 copies of chromosome 13 when you're only supposed to have 2. And it happens because of non-disjunction. This could also happen in anaphase 1, where, anaphase 1 or anaphase 2, this idea of separation here, where you're pulling not the even correct amount. So all of these cells have a problem as a result. So to sum things up, cells follow a cycle to grow and divide, and we have one long part of the cell cycle called interphase, and it has 3 parts, G1, S, and G2. That small leftover piece is called mitosis. That's all about dividing the nuclear division. And in regular mitosis, we want genetically identical cells, and we have 4 sort of 5 stages: prophase, metaphase, anaphase, telophase, and cytokinesis and telophase happen at the same time. If we don't want to make genetically identical cells and we want to reduce the chromosome number, we're going to use meiosis, and there's a meiosis 1 and a meiosis 2. Independent assortment can happen, and that allows us to have genetic differences as well as crossing over, and that mitosis process is not perfect, and it can cause some bad effects by non-disjunction where those errors occur.